Hello everyone. In this video, I'm going to explain the role of nucleosomes to help supercoiling the DNA. What do you think is nucleosomes? Well, the structure of nucleosomes has been shown over here. Nucleosomes are protecting DNA and allow DNA to be packaged, and a nucleosome includes wrapping DNA around itself, and also this nucleosome as a whole not only includes the DNA but also includes some protein inside. So inside this nucleosome and this area includes proteins. What kind of proteins do you think that are in the nucleosome? If you look at this area, you will see the DNA without any nucleosome structure starting from the top, and as mentioned as naked duplex DNA. And then you will see the structure of the nucleosomes which wrapped around the which includes the DNA wrapping around the proteins, some special proteins, and together make one structure of a nucleosome. And then these nucleosomes are wrapping together in order to make another structure which is called solenoid. And this solenoid structure has the 30 nanometer diameter. So this is the diameter of this solenoid structure. And then we will have extended form of the solenoid, and then again the more condensation from the solenoid that makes a structure of a chromatin, and then more condensed, this is a condensed section of a chromatin here, and the condensation of chromatin by wrapping around itself by more supercoiling again, or by more compacting, then it creates the structure which we know it is called as chromosome. Looking at the box over here, you will notice that prokaryotic DNA, they may have supercoiling, but the difference is they are not organized by histones. So supercoiling with histones is the characteristics of eukaryotes and not prokaryotes. And this is one of the reasons, if you go back and look at the categorization or classification of the starting from the top, you will see that one of those differences between prokaryote and eukaryote is that the DNA from prokaryotes don't have histone proteins. Here is the structure of a nucleosome with more details. Now you will see that there are different proteins in order to make this structure as a whole nucleosome. Well, how many proteins are involved with one nucleosome? Pause the video and tell me the number. Yes. You just need to count the number of the protein over here. Can you count? And 1, 2, 3, 4, and 4 in the bottoms, like 5, 6, 7, and the one in the under the 3, that would be number 8. So we have 8 different eight proteins, but these proteins are 2 copies of 4 different types. So basically we have the type of proteins, the 4 types here, and the same as these 4 on there. So we have 2 copies of 4 different types of proteins. One protein that is here, and you see it's the histone H1, and the histone H1 binds DNA in such a way to form a structure called 30 nanometer fiber, which is solenoid. So H1 histones are very important and have the main function in making these 30 nanometer solenoids. Here is a very good question. Why does eukaryotic DNA need to be supercoiled? Pause the video and try to think and answer the question. Okay. Supercoiling is when the DNA strand has been wound back on itself multiple times so that the molecules become compacted. This is what has been written over here. But there are real facts about the reason for being supercoiling. Well, what happens when the DNA is supercoiled? The length of DNA in human, which means in eukaryote, is approximately 2 meters. Eukaryotic chromosomes have like 15 to 85 millimeter in length, each chromosome. So this is the length of each chromosome. So if you imagine that this is a structure of one chromosome, this length could be around 15 until 85 millimeters, because we have smaller chromosomes and we have longer chromosomes. And the nucleus in the eukaryotic cell has a diameter of approximately 10 micrometers. So there are some measurements if this is a nucleus and if this is a cell. So this diameter is something near 10 micrometers. So there are some numbers that maybe it's you don't need to memorize, but it's good to know that what is the diameter of a nucleus, what is the length of a chromosome, and what is the length of the DNA inside the cell in eukaryotic organisms. Okay, going back to the question again, why does eukaryotic DNA need to be supercoiled? Essential to pack genetic material into the nucleus. It is important because the genetic material needs to be packed. And a nucleus, as you go back and you look to the numbers, nucleus is quite small comparing to the total length of DNA. How is it possible that a 2 meter DNA will be packed in a 10 micrometer nucleus? So the only way is packing. It cannot be just go inside without package. To organize DNA to allow cell division to occur, well, that's for sure. To control DNA expression. This is really good and interesting topic, because this is the way that DNA is controlling which part needs to be expressed and which part needs to be available or activated for the next step for gene translation in order to make specific proteins. This supercoiling is very important to allow the process of gene expression, because it allows some parts to be transcribed and it doesn't allow for other parts. Allows cells to specialize by permanently supercoiling DNA. So for some cells, the area might be permanently supercoiled, such as heterochromatin, What happens if they are permanently supercoiled? As you can see, they are going through the specialization and become a complete differentiated cell, and that part of DNA is not allowing anymore for changes. So that's why we have like a kidney cell, or we have a skin cell. So these cells already, their chromosomes and the part of the DNA has been already expressed, and the genes were has been activated before becoming a skin cell, for example, and not after that. So there will be a permanent supercoiling on those heterochromatin on those part of the DNA which is called heterochromatin after becoming permanently supercoiled. So this is very, very nice topic, and I really love this one. And I just need you to understand how interesting is this these two sentences. In the following, again, you will see the importance of transcription regarding those parts of the DNA who are becoming active, which is called euchromatin. So transcription of actin chromatin or euchromatin can be promoted or inhibited by the associated histones. Gene expression is a very nice topic, and these proteins are responsible for allowing or activating or by inhibiting those regions of the DNA that to be transcribed or not. So this is the part that the discussion part of the nucleosome, and I hope that you understood how the nucleosomes are supercoiled. I am putting a video over here so we can watch the structure of the nucleosome and how it will be supercoiled. However, you need to, once you have the slide, you need to click on the link and see what is written in the article to find out more about nucleosomes, and also this link which explains the visualization, and this is very nice. And you will find such kind of beautiful structures in the JMAL visualization website. And once you go there, you can think about these two questions and answer these two questions. Identify 2 copies of each histone proteins, and this can be done by locating the tail of this protein, and suggest how the positive charges help to form the nucleosome with the negatively charged DNA molecule. So these are the 2 questions, and I wish you have the answer or find the answer of these 2 questions after going to research on this website.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
For a cell to develop and function properly, it must turn on the right genes at the right time. Most of the genes in a eukaryote are located in the nucleus, within a tangle of material called chromatin. Researchers are beginning to discover that the structure of chromatin dictates whether or not a gene can be transcribed. Most of the chromatin in a cell's nucleus is highly ordered and tightly coiled. In chromatin, DNA is wrapped around several types of proteins called histones. Each of the many coiled units is a nucleosome. The histone proteins have an overall positive charge, and the DNA has a negative charge. These opposite charges attract, forming a tight association between the protein and the DNA. In this chromatin structure, the DNA of a gene is virtually inaccessible, and RNA polymerase cannot transcribe it. If the DNA is inaccessible, how does a cell turn on a gene? There are actually many different triggers for initiating transcription. As one example, a cell may receive a signal during development that results in the activation or production of a specific type of protein called a regulatory transcription factor. The regulatory transcription factor recognizes and binds to exposed sequences in the chromatin of a specific gene. It may then work in conjunction with a set of proteins that can change the chromatin structure. For example, chromatin remodeling may occur through the function of a type of protein called a chromatin remodeling complex. The action of this complex is not well understood, but the resulting structural changes expose regulatory sequences in the DNA. The regulatory sequences may be close or thousands of nucleotides away from the transcription region of the gene. For simplicity, we'll examine the naked DNA of the gene. About 30 nucleotides upstream of the transcription region is the promoter, the core regulatory sequence of the gene. Next to the promoter lies the promoter proximal element. Promoter proximal elements stimulate transcription through the binding of tissue-specific regulatory transcription factors.
We are not going to talk more on the gene expression at this video, and I'm going to show this video later when we talk more in details about the gene expression. But as you saw, this area are the places and how the mechanism of gene expression is happening by starting from a naked DNA, which you don't see any histone proteins over here, but all the areas of the DNA, then in the future you will notice that how histone protein are responsible for activating, for allowing that the promoter, which is the starting point of being recognized by the DNA polymerase, and all these stuff and all these things will be I will explain later in the gene expression in the future lessons. Thank you for watching.